Functional magnetic resonance imaging is using the exact same technology that we outlined in the previous video. The idea of magnetizing the water molecules in the body, using different resonant frequencies to energize those water molecules, and then taking a picture of them once we turn the radio frequency pulses off. The big difference with functional resonance imaging is that we are actually focused on looking at the blood flow rather than just the anatomical pictures. So really, in a core MRI scan, it takes a lot longer, and the focus is on imaging the anatomy. Whereas functional MRI is all about looking at changes in neural activity. Right? And it's all based around changes in the oxygen flow of the blood. The brain uses about 20% of the total oxygen, even though it's only 1 to 2% of the mass. And there is a complicated mechanism to ensure that there's adequate oxygen supply to the brain. When neurons fire, they release the stored potential energy. Right? Remember, we talked about how the energy in the neurons are stored based on the concentration and electrical gradients of the sodium and potassium. But once the neuron has fired, it has to return back to baseline, and it uses what? The sodium-potassium pump, right? The sodium-potassium pump returns the neuron to baseline so it can fire again, and the sodium-potassium pump takes energy. It uses oxygen. So when a neuron fires, a bunch of oxygenated blood has to flow in. And what the MRI signal is doing is it's trying to detect the oxygen level. As neural activity goes up, blood oxygen level goes up, and the fMRI signal should go up as well. This is called the blood oxygenation level dependent signal, or the BOLD signal. What's important to realize is this is an indirect measure of neural activity. It is not directly detecting neural activity, but rather detecting something that we think correlates with neural activity. If we were to discover tomorrow that blood oxygen level does not correlate with neural activity, you could toss out the last 15 years of psychological research. Luckily, that's not going to be the case. But it is important to realize that it is an indirect measure. We also have to take these maps of the brain very, very quickly, which means that the resolution is very minimal. And so with the anatomical MRI, you can take a high resolution long scan. With the fMRI, we have to take many images of the brain very, very quickly, and the only way to do that is to decrease the resolution. Right? So this sort of blurry cloud is actually the blood oxygen level signal, the BOLD signal, but it is a very low resolution. We can, however, map this sort of fuzzy blob onto the anatomical scan. We can match them up, and we can actually then see in these heat maps where the blood oxygen level changed the most. This is a screenshot from a program called AFNI, which is a freely available program that is designed for fMRI analysis. The way it works is it detects changes in the BOLD signal, which we're seeing here down here in white, and then it attempts to match them to a modeled signal. So in this case, if you think about it as a finger tap, so you're going to tap your fingers for 10 seconds, tap, 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 and then you're going to stop. Right? Well, the area of the brain responsible for tapping your fingers for 10 seconds is going to be more active when you're tapping than when you're not. So if you tap for 10 seconds, stop for 10 seconds, tap for 10 seconds, stop for 10 seconds, you're going to get a predicted blood oxygen level that forms this sort of sinusoidal wave that's going to map the on and off of the finger tap. AFNI then attempts to match what areas of the brain blood oxygen signal maps best to the example, the modeled prediction. And those are going to be the areas of the brain that are most likely to be associated with whatever task is at hand. So we can use the fMRI to literally detect thought. Right? There's some fascinating research that has been done. There's no way to cover even a tiny fraction of it, but here are some interesting ones. This was one that took some control patients and asked them to imagine two people playing tennis. They then imaged the brain and saw the areas of the brain that seemed to be active when they were imagining two people playing tennis. They then took somebody who had been under a coma, non-responsive for years, and asked them to do the same thing. And what they discovered was a very similar match. So the person was able actually to hear the request and recreate the thought in their brain, and they were actually able to see that thought in this comatose person's brain. If you remember the very beginning of class, we watched a video about a guy who was able to identify people's thoughts when they're thinking about specific nouns. Well, his research is now extended in Carnegie Mellon to being able to process people reading specific sentences. You can actually differentiate sentences that are similar in structure and theme, but still different, and can detect observable differences in the brain that lets us predict what the person is reading. In fact, he's even gone further and recently has said that he's able to detect complex thoughts like suicide, and where he actually says he has mapped the suicide scan, right, when a person is suicidal, what their brain would look like compared to a control. So this is an area that is rife for development. It shows a direct connection between changes in the biology of the brain, neurofiring, and specific changes in thought and behavior. And this development of the fMRI is one of the primary fuels for the explosion of neuroscience in the field of psychology over the last few decades.